Hello guys. Okay, so the topic for today is protein synthesis. You're gonna be coloring a little bit, but you're gonna be copying the stuff that's on the red. So let me move my face over here. Let me read this. It says you're gonna have to explain the purpose and process of transcription and translation using the models of DNA and RNA. So here you have a picture of like DNA, then you have RNA, which is a new thing that we're gonna talk about today, and then at the end you end up with the protein. Now, so this is the process of protein synthesis. The word protein is a type of molecule that makes, gives you your muscles and your makes your bone, your skin, and all of that, even your hair. And RNA, DNA is obviously the DNA, genetic material. RNA is a new thing that we're going to talk about, which is a copy of DNA. So when you go from DNA to RNA, that's the step of transcription. When you're going from RNA to the protein, that's the second step, that's translation. And we're going to go into detail. So the first thing you need to do, on your left side, there's a picture like this, but it's white. I need you to color it blue. That's DNA. Okay, you know that's DNA. We talked about it before. It's like two sides, so it's kind of like a little, little squiggly. It's a twisted ladder. It's called double helix. So pause the video really quick, color it blue. Let's move on. So go ahead and pause the video, copy this, and then I'll talk about it. Okay, now that you copied it, it says DNA is found in the nucleus. You know that. That's not new. You have a cell. Inside there's a nucleus. Inside of the nucleus there's the DNA. It's the most important part. Now, the DNA is going to have four different letters, four different bases. It's A, T, C, G, right? Apple tree, cargo rush. The full names are adenine, thymine, guanine, and cytosine. And you also need to know that they are complementary to each other. The word complementary means that they go with one another. So apple tree, right? A goes with T, G goes with C. You did that yesterday. Complementary means they kind of pair up. Now, let's start talking about this second type, which is RNA. Now, let me review DNA really quick. So you have a double-stranded. Don't copy this yet. I'll give you time. So double-stranded, that means that it's double, two sides. I'm just going to put it, two sides. Deoxyribose sugar. It has a deoxyribose sugar. Now, I know before I always said sugar. Like it has a phosphate, sugar, and base. Now we're going to give it a name, deoxyribose sugar. And the reason it's not that hard, even if you're struggling to say this word or whatever, just look at the first letter. Deoxyribose, that's D for DNA. The bases, like I said, the letters are C, G, A, T. Here's a picture of DNA. It looks like a little squiggly line or whatever, a little twisted. Now, for RNA, it's going to be a little bit different. It's kind of the same, but not really. Instead of being two, it's not two. It's only one side. It's one strand. Instead of having deoxyribose, right, there's no D, so it's going to have ribose sugar. Ribo, R, that's for RNA. Now, the bases are going to be C, G, A, and U. Are basically the same, except for the T. There is no T. Instead, there's a U. And we'll practice this on Monday. But then, for now, I just need you to know that there's one letter difference. Their RNA is one strand, and then they have ribose sugar. There are three types of RNA. We're not going to go too much into this today, but one is called mRNA, the other one's tRNA, and the last one is rRNA. And yes, it's spelled like that with the lowercase first. Go ahead and pause the video and copy this now, please. Okay, let's move on. Now, I need you to really quick color the mRNA, right, in your picture red. So it's going to do something like this. Doesn't have to be perfect. Okay, now the next picture is the reason why we colored. I want you to see what's going on. So you have the DNA, which is blue. Then you have the RNA, which is red. The DNA is going to open up, and then from the middle, you're going to get a piece of RNA. So basically, you have the DNA, right? And it's going to really open up, and then from the middle, it's going to come out RNA, which is like a copy of DNA, but it's only one side. It's only one strand. Go ahead and color this little image just like this. The DNA is going to be blue, and the RNA is going to be red. Pause the video. Now that you did that, let's move on. So here's another picture of basically what's happening. The DNA is blue, the RNA is red. Okay, so this is the first step. This is where you're going from DNA to RNA. So that's what it says right here, DNA to RNA. That's transcription. This is the first step of protein synthesis. DNA to RNA, it takes place in the nucleus. Since that's where the DNA is, in the nucleus, this is where this happens. Now, the first step, like I said, the DNA is like kind of like this. Then it's going to open up. So as number 1, it says DNA strands separate. Then number 2, it says RNA nucleotides bind to separate a DNA template strand, forming an mRNA strand. That's kind of a lot. So let me explain using this little image. Look, before I keep going, copy this. If you haven't finished copying this down, please. Okay, now here you have a DNA. How do I know that's DNA? Hold on, I'm just going back and forth. That orange is DNA because it's 2 sides, it's 2 columns. Now the purple is going to be the new one that's being formed. That is RNA. How do I know that's RNA? Well, 2 ways. It's only 1 strand, instead of being double. It's not double, it's only 1. And also, you see the sugar. The sugar on the DNA is the orange. It has a D for deoxyribose. But the sugar in the RNA has an R for ribose. So that's 2 ways to see it. If you see 2 strands, DNA. If you see 1 strand, it's going to be RNA. If you see deoxyribose, that's DNA. If you see ribose, which is the R, RNA. So basically you understand what's going on, right, so far? Here's another example. DNA separates, right? They were together. They separate. Then you have little pieces that come and they start attaching, and that forms an RNA. Okay? Now, this is the next step. I need you to go ahead and pause the video, color the color that they say, or just like this. The RNA is going to be the one at the bottom. It's just a single strand. And then the amino acids are going to be green. The amino acids are the little balls. Okay, pause the video, copy this exactly like that, or color, I mean. Okay, so one thing to mention, you have this thing, okay, this little, it has like 2 pieces, one at the bottom, one on the top, and that's called ribosome, and it has the name right here. The ribosome is going to, it has like a little hole, okay, and the RNA is going to come and it's going to go in and it's going to start doing things inside, kind of like a little factory. So these are the instructions. This is going to come in and it's going to be connecting here. Now, every time it connects, these little things, that's called tRNA, we'll talk about that later, this little ball is going to be dropped off. So when it drops it off, it adds it to the chain. So it adds it to the chain, and this chain is going to get longer. So let me go back to what do we need to know this kind of thing. This is, imagine your hair, okay? You have DNA in your skull, right, in your head. You need to make more hair, so all that's going to be happening is the RNA is going to come, and it's going to be dropping off a little piece, and it's going to be adding to your hair. So your hair is going to get longer and longer, it's like a chain. Go ahead and pause the video here, and copy that down, please. This is the next thing. Okay, so this here says, this is the second step, when you're going from RNA to protein, which I already kind of explained, but I'll explain it again. You have this thing that is like a double thing, kind of looks like a, I don't even know, like a little mushroom. Okay, so this is the second step. I already kind of explained it, but I'm explaining it again into more detail. So this is the second step. You have this little ribosome here in the back. It has that like a double shape, right? Like this. It kind of looks like a mushroom or something. So the second step is called translation, right? When you're going from RNA to protein. Translation is the second step in protein synthesis. When you're going from RNA to protein, it takes place in ribosomes. The mRNA attaches to a ribosome. Okay, so the mRNA is going to come, that's the single strand, the line is going to come into the ribosome. Then you're going to have the transfer RNA, which are these red squares, and they're going to carry the correct amino acid to the ribosome. An amino acid is just a little ball. They're going to come and drop it off into like a sequence. The ribosome is going to bind the amino acids together to make a protein chain. So the ribosome is going to kind of like get the little balls, put them together, and scoot them over to make a long chain. That is called a protein. A protein is just a long chain of amino acids. Here's an example of the second step. Second step happens outside the nucleus, right? This happens in the ribosome. So you have mRNA. How do I know it's mRNA? Because it's only one side. It's going to leave to that little mushroom looking thing. It goes inside. That's a ribosome. Okay, and then it repeats. We're going to watch the whole process all together. It's going to take a little while. This is the first step, transcription. Okay, you have the DNA opening up, then you have little pieces that come and attach to the DNA to make a new thing called mRNA or RNA. You know that's RNA because it's only one side. Once it's ready, like it's ready in the nucleus, once it's ready, it's going to leave the nucleus. You're going to see like right there, that's the nucleus. It's going to leave it outside. It's going to go outside. And then you have the little mushroom, that's the RNA. And it's going to go and start connecting. Now, every time it connects, it's going to leave, but it's going to leave that little ball on the top. In this case, they're not balls, they're little squares or whatever. But yeah, it comes and it connects based on the code, and it drops off a letter, a thing. So it connects, leaves. Then once it's done, the ribosome leaves, the mRNA leaves, and you end up with a protein, a chain of protein. Okay. Here's another example of how it looks. You have the little ribosome in the back, the purple one. You have the little tRNA that are coming to drop off the little balls connected to the mRNA, to the code. So you see each one comes, connects, and drops the little ball. And then that's pretty much it. You end up with a chain of protein. Okay, so here's the overview of everything. So you start, we're going to talk about this picture later, but you start with DNA, which is double. Then it opens up, and it starts making that black line. That's RNA. This is the first step, okay? When you're going from DNA to RNA, that's transcription with a C. And then you have the little, whatchamacallit, RNA here. It's just, again, it's just like, you know, first step, second step, third step. Then fourth step, you're going to go outside. So transcription is done. When you leave the nucleus, transcription is done. Then you go into translation with an L, translation. The RNA is going to leave, and it's going to go into this little mushroom-looking thing. And then you start dropping all the little balls, and this chain starts growing. This is called a protein. These little dots are called amino acids. Okay? If you're still confused, make sure you ask me. We're going to be adding more things to this starting Monday. All right, thanks.